Very good afternoon, everyone. Swagat, Maraba, welcome. We are very excited to have you here. And again, thank you so much for taking the time out to attend this invigorating and a very interactive session, hopefully, on designing engaging courses using videos with a very special focus on cell and molecular biology subject areas. I'm Prerna Bhanushali, Manager for Customer Success Team at Jove. And joining me are my colleagues Tani and Preeti. They are from the subject matter expert team, and both are PhDs in their field of interest. I'm very delighted to have with us and to introduce our guests for today, Dr. Sudhir De, Dr. Sneha Bhogle, and Ms. Purnima Ravindran. They're scientists and teaching faculties at Indian Institute of Science and Education and Research, Pune. That's ISA Pune. Their research study focus groups are on the ecology and evolution of population with an emphasis on how organisms interact and impact various environmental aspects. Their labs also conduct a lot of workshops on molecular biology to cover concepts and techniques to help train young minds and students with a hands on deck approach. Coming to the theme of today, we will first be dipping into the minds and experience of our guest speakers and learn from them the engaging pedagogical ways they have been implementing to teach and train students. Next, my colleagues and I, we will cover a little bit on the overview of Jove and the scope of content that Jove offers on the subject. And finally, we will also cover on how to make all of these contents available to your students. Should you be a teaching faculty or a PhD scholar who's a teaching assistant, we can bring everything together for you with very little hard work from your end and just pick and choose. So here is the liberty to be spoiled. Over to you, Professor Sudhir De and your team.
Thank you, Prerna. I will start sharing my screen. Let me know you can see my screen.
Yes, it's visible.
It's visible, right?
Yes.
Is it now visible in the presentation mode?
Yes.
Excellent. Thank you. So thanks, Prerna and everybody else at Joe for letting us connect with students and faculty not only in India but also abroad. So today we're going to talk about the series of workshops on molecular biology that we have been conducting at ISA Pune since 2019. But before we get there, I would like to quickly talk about what are the major issues that one faces when teaching, particularly molecular biology in the context of a country like India. And perhaps the biggest problem, prior to that, we need to know why should one do that. As we know, molecular biology techniques, they are very critical for answering fundamental questions in many areas of biology, as well as being very applied stuff in industries like pharma and food and bioprocesses and so on. And therefore, it is important that students in every subdiscipline of biology have a very good understanding of these techniques. However, when we come to actual details of how to impart training in MolBio to our undergrads and postgrads, we actually face quite a large number of issues. And the first issue, of course, is that molecular biology is really, really expensive. I mean, if you have ever done an experiment in MolBio, and have had the luck of using antibodies, you know how expensive they are. And also the machines that one requires for doing MolBio, again, they can really set you back by a large number of dollars, rupees, dirhams, based on wherever you are. As a result, whenever you go to a college or an university, typically it becomes very difficult for such places, unless it's a very rich and very elite kind of a place. It becomes very difficult for them to provide facilities for every single student to do every single step. As a result, because of large class sizes, etc., in most places, what you have as small biopracticals are essentially demonstrations. The teacher stands in one corner and performs the experiments, and the students are expected to, be around them and figure out what's going on and then just keep it in their mind. More critically, whenever you have a college or a university, lecturing or tutoring schedule, most places you have very modular periods or routines as we call them, and therefore the best that a college can give for a practical exercise is maybe about 3 to 3.5 hours, and hence the instructors are forced to push everything, shoehorn everything into that available amount of time. Now, those of us who have done molecular biology, we understand that this kind of a shoehorning in terms of time is very, very not suitable for molecular biology work in particular. And therefore, most of the time one has to break the thing down into modules and therefore lose a sense of continuity across the day. And finally, this is more related to the pedagogical aspect of it. In most places, molecular biology exercises are taught as an extension of the theory. So you learn PCR in your theory classes, and then you go to the lab and you perform the PCR. Now, whenever you have this kind of a teaching, then the focus mostly is simply on getting the right result, getting the right band. Okay, rather than using molecular biology as an object of inquiry by itself, a thing in which even when you are committing errors, you are learning a lot. All these things makes proper training in molecular biology, particularly the practical aspects of it, a big challenge. And as a result, as I said, most students did not get to do the experiments on their own. And most of the times, in the minds of the students, molecular biology is essentially a bunch of techniques. Okay, it is analysis chain electrophoresis, it is PCR, it is Western blotting, etc. Not really an integrated whole which can be brought upon to bear on a problem. Because of this, as I said, most of the time practicals are viewed as some kind of an ideal exercises where a certain result needs to be obtained, and not really treating them as an inquiry driven thing which allows one to learn even from their errors. Because of this, most students often find the practicals to be rather boring, rather pointless, and when these students then finally go out in the world, either as a researcher or as somebody who has to work in the industry to solve some industrial problems, then most of the times they don't really know what to do, because the picture that of molecular biology that they have in their mind is very difficult or very different from the use cases that they are suddenly trusting. So, in order to see whether molecular biology can be done in a slightly different way, we came up with a series of, a program which conducted a large number of workshops in molecular biology at ISET. We have been doing this since 2019, and we've been doing it for school students, college students, postgrads, PhD students from all over India. In fact, a few students even from abroad. Now, obviously, as I told you, molecular biology is instrument heavy. So where did we get the instruments from? So in order to get the instruments, we actually went to various corporate houses of India, and through their corporate social responsibility, they gave us different amounts of funds at different times, one-time donations, using which we brought our instruments. And of course, whenever you are doing mol bio, the teaching is extremely important, and you need to, you just cannot take somebody from any area and just say, okay, now you are going to teach molecular biology. You need dedicated faculty. So we got two full-time staff, and I'll introduce them to you in a minute. And their salaries were entirely CSR funded based on yearly donations. The infrastructure is what our institute, ISIS Pune, gave us, and the consumables, which are a huge chunk of the expense, that we raise from the participants on a no profit, no loss basis. So these are the administrative aspects of what we are doing. The reason I'm talking about them is because there are many faculty over here who might want to think if how to get such a program happening at their place. So this is how we did it at our. So these are our various sponsors. Weapendorf, High Media, Bartleby, these were the people who gave us money to buy the instruments. I know Eppendorf also gave us money in the initial 3 years for our teaching staff, their salary. So these two, Praj and Nag, they are our current sponsors, and we have been partnering with Joe for the last 1 year or so, and I'll talk a lot about how we have been doing. So this is our team. So this is me. I'm a professor at the Department of Biology at ISEF Pune. And this is Purnima Ravindran. She is a BSc and a Masters in Botany from University of Pune. And she has been at ISEF Pune for the last 8 years teaching primarily molecular biology. This is Dr. Sneha Pugle. She's also a teaching associate under this program. So Sneha did a PhD from ISEF Pune, then did a postdoc in the US, but then she realized that teaching is her true calling. So she came back from the US and then joined this particular program. So this is our lab setup. Now, this is not the whole lab, but just one overview just to show you how the thing looks. It's a very tiny room and we can fit no more than 10, 11 people in this. And what is most important are the pedagogical aspects of our workshops. I'll quickly talk about them. So these workshops that we do, they are 1 to 3 day long workshops. The basic ones are about 1 day long. The mid or the advanced ones are 2 to 3 day long things. In these, we get no more than 10 people per workshop. Okay. Whenever you are organizing a workshop, typically the idea is you should bring 50, 100 or more number of people. Again, it becomes important in a country like India where the population is so large. However, we said that that doesn't work when you are teaching something as intricate as small bio. So we put a cap on maximum 10 and we just don't go beyond this. And because of this, we are able to ensure that every single participant performs every step of the experiment. And the concept or the focus is simultaneously on the concept underlying the experiments, the design of the experiments, why you are doing what you are doing, what will happen if you forget to, let's say, add chemical A or let's say add excess of chemical B and so on. And finally, a lot about troubleshooting. So anybody who has done all bio knows that many a times you don't really get your desired or your expected result, but figuring out why you haven't got that tag or why the city value is not as high as it should be, that is part of the challenge, and we spend a lot of time doing it. So one major thing as a matter of policy that we do over here is there are no cost cuttings in this particular context. We use the same consumables from the top companies, we use the same machines from the top companies that are used in our own research labs. And therefore, at no point do we use, let's say, machines whose readings are not dependable or they're not robust. And of course, the biggest thing is that we have two dedicated people, Sneha and Purnima, that I talked about, who are doing just this and nothing else. So, what are the various things that we cover in our workshops? So, these are some of our DNA related techniques. These are some of our RNA related techniques. So, you can see that these are, qPCR steps over here. Then we do a bit of cloning, we do a bit of bioinformatics, simple introductory bioinformatics, and then we also do a lot of protein related techniques. And next year, if everything goes all right, we are planning to introduce things like CRISPR-Cas9, ELISA, and SSR marker-based plant genotype. However, these are just names and nothing else. The how we actually go about making them interesting for our students, that is what Sneha is going to talk about now. So Sneha, over to you.
Thank you, Sutirth. I just share my screen now. Just a second. Is the screen visible?
Yes, Sneha, you are visible.
Okay, great. So thank you. So throughout the year, we conduct a variety of different molecular biology workshops, which are divided into two categories, basic workshops and advanced level workshops. So under the basic workshops, currently we have these three workshops, one of DNA fingerprinting, the other of DNA-based species identification, and a workshop focused on the enzyme catalase. While in advanced workshops, we have regulation of gene expression, qualitative and quantitative, a workshop on TA cloning, and a protein workshop. Apart from these hands-on molecular biology workshops, we also conduct some demonstration workshops. So in the last year, we conducted 2 demo workshops, one on sandwich ELISA and the other on dot ELISA technique. So when we conduct these workshops, we don't consider the molecular biology techniques that we cover as individual entities or in isolation. We give context to these techniques. For example, if you consider one of our workshops, microbial investigator DNA-based species ID, we present our students a real-life scenario. For example, it's like we have a disease outbreak in our state, and the investigators have understood that it's a bacterial disease, but they have not identified the bacteria. So what do? They send the samples to National Institute of Virology in Pune. And now these students, we are the scientists, and now it's our job right now to investigate or identify the bacteria from these samples. So now the students understand that this is what we have to do. We have to identify the bacteria using molecular biology techniques. So what should we do? So the first step that they do is they want to isolate the genomic DNA from the bacterial samples. So automatically, the first step that comes into play is you have to break open the bacterial cells because genomic DNA is present inside the cells. So they get hands-on experience with the entire DNA isolation process, how the chemicals are used, what is the function, what is the significance, and so on. But then what next? You have to check whether your DNA isolation was successful or not. What is the quality of your sample? For that, they get introduced to the technique of agarose gel electrophoresis, wherein they load their own samples and they figure out how they have done with their DNA isolation protocol. But this does not still solve the question. Do you identify the bacterial sample based on its genomic DNA alone? No, there is one such strategy where you can focus on one gene rather than the entire genome. So how do you amplify that gene? Then comes into picture the role of PCR, where you can amplify a certain gene. And again, whether this PCR has worked or not, the students have to again do agarose gel electrophoresis to check their PCR. And now the PCR product that you have, you sequence it, and the sequence that you get, you use that sequence in a bioinformatic tool called BLAST, which does sequence alignment, which finally gives you the identification of that particular bacteria. In our workshop, we do not conduct the sequencing step, but we give them a representative sequence, which the students take up and do the blast themselves and finally identify the bacteria. So now, instead of working with these techniques separately, they have a question in mind, they have a flow, there is a logic to it, and the students get more engaged and interested in such kind of a workshop. Moving ahead, I would like to discuss another example with respect to our advanced workshops, which is the qPCR workshop. So here again, you can see these techniques of molecular biology, which are maybe not even done in practical. Some of them are done only in theory. And so the children or the students don't have an idea about the implication and the actual application of these techniques. So in this particular workshop, which is the qPCR workshop, we again give our students a real life scenario. For example, we tell them that we are working in an industry and the current project that we have is to quantify the level of beta galactosidase in bacteria which are grown in different environments. So the students immediately understand that they are now working with a single gene and they have to understand how environment plays a role in expression of this particular gene. So if you want to do a qPCR, you have to do primer designing, right? You want to amplify this certain gene, you want to quantify it. So we introduce the students to detailed in silico primer designing with a special focus with the qPCR primers, because that's slightly different from the regular PCR primers. So the children get exposed to this exercise as well. Okay, so now they have the primer design with them. They have the primers. What next? You cannot immediately go to qPCR because now they understand that it's the expression. You cannot use genomic DNA as you see in the previous example. You have to go to the RNA, which again is a problem because RNA won't be recognized by a qPCR system. So they have to do this different or additional step where they convert the RNA into its cDNA form, and the students do this cDNA synthesis themselves. They do these reactions and they use the cDNA as a template for your qPCR experiments. But again, moving directly to qPCR is also not correct. You have to standardize or optimize your assay, show that it is working at near 100% efficiency. Only then you can move to the actual qPCR assay. So all these things, students get hands-on experience and they also understand the importance of it, after which they actually get CT values. But then again, using or having CT values alone doesn't answer the question that they started out the entire workshop with. They have to analyze the data, where in our current workshop, we show them the data analysis using one such method, delta CT method, where they know how to use this, they quantify their own data, they get relative gene expression graphs, and we also focus on the statistical part of it so that the students understand whatever data they have gotten, is it statistically significant or not? So even in this advanced level, the students get a complete idea from the start to the end. And in this entire process, they get hands-on experience and a better conceptual understanding of all these different molecular techniques that we have just covered. So apart from hands-on being one of our major focus, we also heavily discuss, or we also heavily have other focal points like the part of troubleshooting. Now, if you take this gel picture, for example, all the three students have done successful genomic DNA isolation. But if you see the sample I done by student number 2, the genomic DNA is shared. So here we don't just leave here that, okay, the genomic DNA is isolated, everything is great. But we talk about this particular sample as in why did the shearing occur? Where were the errors introduced? How could we change or reduce or completely avoid these errors if we repeat this? So the students have a better understanding of how to read their data so that if there are any errors, they can do a better job. Another example I would like to give is with respect to qPCR data. So if you see these three graphs, they are done by one student and they are replicates of each other. So 3 replicate samples, of which this one replicate is giving a completely different CT value. So this completely results in a different data interpretation for that particular student, and it's a clear error. So we again talk about the fact that where did this error get introduced, how we can change or reduce or avoid it, and how can we better our experiment. Apart from troubleshooting, another focal point is experimental design, as I mentioned before, the concept and the context that we give to it. But during our experimental setup, our students also work with controls. So not just their test samples, they also do these controls. For example, when they set up PCR reactions, they work with negative controls and positive controls. When they set up qPCR reactions, they not only work with the gene of interest, but they also work with the normalization or housekeeping genes, which is a very important component of a real-time analysis. Or if they're doing any enzyme assays like enzyme kinetics or enzyme specificity assays, they perform these assay controls like enzyme blanks or substrate blanks and so on. So all these things together give the student an idea about how an experimental design should be, which makes your data valid and acceptable. And lastly, we focus again heavily on data analysis and interpretation. So students generate data after the experiments, but how to make sense of this data? It can be as simple as reading a gel, whether it is a DNA gel or a protein gel, with respect to presence of specific versus non-specific bands, uh, size of this particular product, and so on. And we also expose the students to different tools for data analysis like sequence alignment tool like BLAST, or we use ImageJ software for calculating the migration of a DNA sample, or we also use very high-end softwares like the one we use for qPCR analysis, which gives something like the amplification curves, the melting curves, which help in product specificity, standard graphs. So basically, the students not only just create data, they are also aware of what to do with this data, what tools are available, what resources are available, so that they can make a better analysis and interpretation of their own data. So together, by applying all these things, what we achieve is students get engaged and they are interested in what they have done, and they get a better understanding of the concept as a whole rather than studying them or performing these experiments individually. So with this, I end my part, and I would like to request my colleague Purnima to present the next part. Yeah, thank you, Sneha. Now that we've heard both about how the idea behind starting of the workshops as well as how the workshops are actually conducted, let us go to some of the impacts that we can see after 5 years of running these programs. The first thing we would like to talk about is how Jove integrates with our workshop. So in January 2013, the Jove team invited us to collaborate with them with regards to sharing video content, which Jove already hosts. So we started sharing these videos with our participants who joined the workshop. There are two different aspects that we cover in our workshops. One is the conceptual aspect, and the other part is the hands-on aspect. Hands-on is what we take care of while the workshop is happening. For the conceptual aspect, we take help of the Jove videos in two ways. The first one is the first one is to reinforce what we are teaching. So while teaching, we may not really have a lot of time. We focus a lot on hands-on, so there's not a lot of time to go very in-depth into theory. So as a result of this, for theoretical reinforcement, the Joe videos are very, very useful, especially the education ones, like you can see here. So this is, for example, one module we designed for the catalase enzyme module. And here you can see that these are the videos that we share with the students. So this is like a reinforcement to the theory part of it, wherein if we have, because of lack of time or paucity of time, if we have left out anything, that also gets, covered here, and also reinforcement of the theory. The second part of how Jove integrates is to appreciate the theoretical background and applications. So we know that we cover a lot of the theoretical ground, and some applications we do cover during the workshop. However, there may be something beyond the scope of the workshop. For example, while doing qPCR, we have an example. We share this video of rabies diagnostic, diagnostic RT-PCR. So they know that, okay, this is a technique we are learning today, but it is actually applied in real life for various diagnostic processes. Already students are very aware because of the corona virus pandemic about the RT-PCR. So they're very interested in this in any case. So these come from the research video portfolio of Jove that we include in our playlist. After this is done, we generally share it with the students. So there is a Jove classroom that we create, where you can see that this, for example, this classroom is for TA cloning, which is one of the modules that we conduct. So we have a series of videos that we collate. We select from the Jove portfolio, we collate them, and with the help of the Jove team, we open these videos and we share this link to this classroom with all the students. So when the students go back after the workshop is done, they can at their leisurely time, they have a time window wherein they can go and open all of these videos and watch them as many times as they want. So for example, these are the videos which are part of the TA cloning module that we have created. Now, this is what we do. After this data is gone to the students, obviously, we wanted to know how much they are making use of this Jove video that we are sharing, platform that we're sharing. So we conducted a very short survey, and we asked them about, what did they think was the usefulness of these videos, what was the interest that was generated while watching these videos, and how informative these videos were. And we got a very good response for the videos that the students have watched. So for informativeness, interest, and usefulness, everything has been rated above 4, where our scale was on a scale from 0 to 5, where 0, where they did not watch at all, which all the participants who answered have all watched the videos. Nobody who did not watch has answered. And we the highest scale that we have is very high. So between 4 and 5. So we are definitely, the videos resources have been extremely useful, and I think the students have been very happy with the resources that were shared via Joy. Now we come to the actual impact of our workshop since the past 5 years. We started the workshop series in 1920, as Sutit mentioned. The first year we had a very good number turnout of students, but afterwards, obviously due to the corona pandemic, we lost out on a lot of workshop days because we did not go online. The idea of workshops was that it has to be conducted offline, it has to be hands-on, so we never went online. As a result of that, our numbers have suffered for the year 2021 and 22. However, once the pandemic restrictions were lifted up last year, we started the workshops full-fledged again with 10 students and 2 instructors. So slowly, gradually, I think we have now reached a level where students repeatedly come for the workshop so that they can actually attend as many modules as possible, even though there is a charge of there is a small payment that they make. However, even then they would like to attend, and they do attend a lot of the workshop modules. In a single year, we have found students repeating as many as 7 modules, all different topics, but they have repeatedly come for the workshops conducted at ISO. So the students who are coming, the demographic is from all over India. We have students coming from all parts of India, smaller cities as well. We have students who have come from places like Akole, and smaller parts in Maharashtra as well. The overall, we take a anonymous feedback after every workshop is done, which is handwritten. After rating these anonymous feedback, we have found that more than 4.5 is generally the average of every workshop that we conduct. So overall, the students are obviously satisfied, and they find value addition with what they are doing in the workshop. And besides that, we have also asked an independent agency, which is called as the Pune Knowledge Cluster, which is a Government of India initiative. They conducted a survey independently last year, and they found that overall the students found the impact of the workshops and how useful they were in their curriculum was 4.57 out of 5. Besides this, last year we also conducted one TA cloning workshop, which was for Praj industry employees. So Praj is one of our sponsors for this current year, and they decided to send a team of scientists who work with Praj for a number of years to come and do one of the workshop modules with us. And it was a great experience for us, and I think they also had a great time, and we got good feedback for this workshop as well. So with this, to sum it up, I think I would just like to say one thing, that our workshops go hand in hand with the curriculum that the colleges offer. And as a result of this, the students, I feel, are better equipped in dealing with the challenges that are real life challenges that they have to face when they go outside after their studies are over. This is a screenshot of the website, of our website, and that is a QR code which will lead you to the website. Thank you so much.
Thank you so much, Dr. Purnima. And thank you, Professor Sutirth, Dr. Sneha, and Ms. Purnima for the wonderful explanation of your workshop. It's very extensive, very hands-on, makes me go back to my teaching and also my student days where we used to keep hunting for these in hands-on deck workshops. So if you were then over there, I would be in one of your images. Awesome. So moving forward, and I would just quickly like to share my screen. I'm hoping you can see my screen. So moving forward, I'm going to take you through a little bit of an overview of Jove and quickly hand it over to my colleague for more in-depth conversation around the scope of content. But before that, I just want to throw in this word mix. Now, what does this word indicate? Because we have now 30 minutes into this webinar, we are primed for academic perceptions. So mix for some of you might feel like stirring. Mix for some of you might feel like pouring two liquids together. Mix could also mean for some of you picking up a dropper and then putting something together. So mix can mean a lot of things, but for an experimental reproducibility, the correct perception of mix is very important because sometimes the meaning of the word or the action of the word might change. So that brings us to Jove and how visualizing, how capturing videographs of research experiments being done, how that really helps increase reproducibility, how that helps increase retention, and also helps in learning outcomes. Very quickly about Jove, we are a world leading producer of scientific videos. Most of us on Team Jove are actually, from the academic backgrounds, PhDs, masters from different fields across the STEM area. We have 18,000 plus videos with video resources. All of them are focused either with high impact animations to explain a fundamental concept or a videograph of scientists actually performing those concepts, bringing them to real time science. We have 1,200 plus subscribing institutes globally, and this is just a peekaboo of what our subscribers look like. So things that Jove addresses, we've already seen this resonate. It helps with for researchers understand the protocol, the way the methodology flows, and increase the reproducibility. It helps training and conducting a review experiment, for example. So if you are already primed to a particular reaction or a particular step in a methodology, you are more likely to perform it accurately. So it helps in saving your consumables. It helps saving your time in trainings. And while the experience is amazing, we would love to build on those training sessions. And next we have, if there is, certain lab themes that you follow. So you can have Joe videos addressed to your lab themes, and then, building a SOP around those lab themes could also be easy. We also help with education side in terms of engaging with students. We also help, for example, if adopted in a flipped learning format, students can come primed to certain information and concepts and come back with better test scores. This is just a few examples of like case studies that show the huge impact of Jove resources. We've already seen it with the presentation that preceded, and here is like a 89% jump of test scores, post watching videos. This is a better performance for the pre-lab and post-lab assessments. So without further ado, I would like to get this over to Preeti, my colleague, for scope of content, and her focus will be on cell and molecular biology.
Thank you, Prerna. Thanks, Professor Sudhit and team. I felt so nostalgic. You just took me to my PhD days. I'll just quickly share my screen. I hope it is visible. Okay.
Yes.
All right. Good afternoon, everyone. Thank you for joining us today. I'm Dr. Preeti Sridharan, subject matter expert here at Joe, and I'm really excited to be your guide as we explore our collections, core molecular biology and cell biology in this webinar. So, to quickly introduce our Joe platform, before we take a detour of our Joe website, so we have two different collections. One is a research collection and education collection. I'll be going through this when I take you to our website on deck. So let me quickly introduce you to our core collections, cell biology and molecular biology. So these collections are specifically designed to simplify any complex concepts that are dealing with cell and molecular biology. So these collections, they have unique and engaging videos that explain the basic of life at both molecular and at cellular levels, and they serve as a dynamic tools to empower the students in their learning journey. So a gist of our core molecular biology collection, which is a classic collection of Job with the 20 chapters and 298 video lessons. They also have a voiceover and subtitles in 12 different languages. So this will really help the international students or non-English speaking students to grasp the concepts in their own native language. And additionally, we also have given additional supplementary videos of scientist in action videos. So these are some of the advanced supplementary videos that are handpicked from our journal collection that discusses the real-time applications in a research setup of any particular concept that is being discussed. So I'll be showing you these videos while we go through our website, and the average play time of each of these videos in this particular collection is 90 seconds. So that is one of the major advantage of our core collection, which is a short length video. So next is our core cell biology collection. So this is also one of our expansive collection with 43 chapters and 61 video lessons. We also have voiceovers here in 12 different languages, and the average video length is 90 seconds. And we also have 129 scientist in action videos here for core cell biology. So these two collections are, thoughtfully crafted to serve a diverse audience, which are mentioned here, and they are such a valuable educational resource across different academic levels and interests. And both these collection, they just seamlessly align with the syllabi of first year and second year biology students, pre-medical students, and then allied health science students, of course, masters and honor students. So of various background, various discipline. So these are also our resources to refresh the fundamental concepts that they have already studied either, or they are just studying in their present curriculum. So apart from this, these collections are also very much helpful, and it also just rekindle the curiosity of any molecular and cellular biology researcher. So as I mentioned, as you can see that there are we are covering a wide range of audience, because these videos are just tailored to align with the global syllabi across various territories. So please just quickly walk with me as I take you through our molecular biology collection with in depth. So to easily understand the variety of contents that we are covering in this collection, we have just divided it into 6 sections based on the broader area where they fit in. So you can find the chapter names and the numbers that are being mentioned in the brackets, or the total number of video lessons that are being covered in each chapter. Um, so coming to one of the most important aspect of molecular biology, which is the biochemistry of macromolecules, we have chapter 1 to 5 as mentioned here. These these chapters, they just focus on biochemistry and structure of macromolecules such as DNA, protein function and structure. We have video lessons on DNA double helix, and then DNA packaging within the chromosomes and different types of chromosomes. So these videos just cater to learners that are at all levels, that simplifies the complex concepts for the better understanding. So next is our central dogma section, which is also one of the important sections of molecular biology. When it comes to central dogma, we are covering these in chapter 6 to 11, that are devoted to the concepts and mechanisms of DNA replication, transcription, translation, and we also have video lessons on DNA repair mechanism. And to be specific, we have I would encourage you to watch this specific video from this section, which is a homologous recombination video, as we know that it is a complex concept and it is a bit difficult to explain that to the students. So if you could watch this video, you will understand how crisp the animation is, particularly for this video. Next, we have for genetics, we have chapter 12 and 13 that discusses the classical Mendelian genetics and gene evolution, featuring video lessons on some of the Mendelian crosses, and then exon recombination, phylogenetic trees, and more. So next is one of the important concept of both molecular biology and cell biology, that is cell signaling pathways. We have one entire dedicated chapter with 14 video lessons in it. So that engages the concepts of cell signaling pathways, including video lessons on some of the complex pathways and some of the most commonly, uh, complex pathways that are being discussed in any textbook if you take. We have video lessons on G protein coupled receptor, Notch signaling, hedgehog pathway, WNT signaling, and more. So you can, I would specifically say this is one of the highlight of this entire collection, because it showcases how advanced concepts like these are being simplified with our animations and a very good voiceover also. So next, on techniques, we have 2 chapters, especially for molecular biology techniques, chapter 15 and 16, that demonstrates both classical as well as modern techniques that are being studied under molecular biology. We have for classical techniques, we have video lessons on electrophoresis, PCR, sequencing, different types of sequencing. And for modern concepts, we have video lessons on CRISPR, gene therapy, and then experimental RNA. So these are some of the video lessons that I would suggest you to watch. And these chapters, they lay a strong foundation for theoretical knowledge and principles of some of the widely used molecular biology laboratory techniques. And finally, we have covered cell cycle, proliferation, cell division, from chapter 17 to 20, that provide a detailed explanation of different aspects of these topics. And including, we have to specifically mention, we have video lesson on cyclin kinase activity, which is one of the important component of cell cycle and regulation. And we have, for cell division, we have mitosis and meiosis videos. And then apart from that, we also have a foundational videos on cancer as a separate chapter here. So overall, our core molecular biology is a go-to resource, I would say, for teaching, that is offering insights to deepen the understanding of molecular foundation of life. So to quickly move on, let's quickly move on to our next collection, which is called Cell Biology. As I mentioned, it is a huge collection with 43 chapters, and this is also relatively a newer collection that got released last year. So for chapter 1 to 11, as mentioned here, they focuses on uh, essential components and functions of life. So these chapters, they just bring fresh perspectives on cellular metabolism and the biochemistry of cell. And this particular collection of chapters has already been proven to be highly beneficial for any biochemistry courses, biotechnology courses, as they offer new engaging content for their students. So next we have chapter 12 to 18 that delve into the fascinating world of membrane biology, covering the aspects of membrane structure, transport mechanisms, protein sorting. And this is also one of the unique section, and I would like to highlight this particular section from this collection, because they have video lessons on active transport, passive transport, ion exchange, and intracellular transport. So it forms the basis of understanding this fundamental concepts of cell biology. So next we have cell energetics, chapter 19 and 20, that are dedicated to cell energetic, that unravel the mysteries of mitochondria and chloroplast and energy production. And this section contains video lessons on cellular respiration and photosynthesis, both in detail. So next is chapter 21 to 31. They take on the intricate topics of cell signaling and cell architecture. So here, I mean, we do have this in our molecular biology as well, but then these cell signaling chapters, as an extension, you can find cell to cell interaction videos, and also cell signal transduction videos. Those are all some of the additions in this particular collection. And apart from that, we have cell movement videos, cell polarization, some of the components that are involved in extracellular matrix. So these are some of the complex concepts in cell biology that are being covered here in these chapters. And for those who really want to reinforce the practical knowledge of cell biology, they can refer to chapter 32 and 33, as they review common laboratory and visualization techniques in cell biology. So some of the videos to mention here, we have videos on animal cell culture, cell separation techniques, and different types of microscopy. So as we all know, these are all some of the essential resources for any cell biology researcher, and these are some of the foundational videos that you can find in this particular collection. Uh, and to conclude, we have chapter 34 to 43 that explore the cell regeneration, proliferation, and repair mechanisms, and delving into the cell division and cell death. Particularly to mention, we have two in-depth videos on apoptosis and necrosis. So along with that, we also offer foundational insights into the exciting field of stem cell biology and regenerative medicine. So to conclude about our core cell biology collection, which is not just the assembly of lessons, but it is also a dynamic and accessible tool that transforms your teaching as well as learning experience. So I invite you to explore this rich resource in our website, and I'm very confident that it will enhance the quality of cell biology education overall. So additionally, apart from the core collection that I showed, we also have science education collection. So this comprises comprehensive videos in cell and molecular biology under basic biology and advanced biology sections. So I'll be showing you how to reach these resources in our website shortly. Um, so I would also say that these videos are or a perfect blend of both theory and practice. So the uniqueness of this collection is that they are longer duration videos with an average watch time of just 8 minutes in them, and also they have a timestamp. I'll be showing you all these in the website, and this is how our typical science education video looks like. And apart from that, we also have lab manual videos. I'll be showing you these two collections. So let us now reach our website. I'll just quickly share my screen to show the website. So you can reach our website by accessing www.jove.com. That will redirect you to app.jove.com. So I would first highly recommend you to sign in using your institutional email ID. If you have not created an account yet, I think Prana will be taking you through how to create an account and access our resources. So yes, once you sign in, you can find our two resources here, research and education. So under education, I'll first quickly show you research resources. So we have our video journals here and our encyclopedia of experiments section here. So these are the collections that we have under encyclopedia of experiment. These are some of the advanced research experimental videos that are from our journal collection that will have both it's a combination of again advanced concepts and the how it is being performed in a research setup. So I can just show a video here so that we yes, this is a collection on encyclopedia of experiment. It will be very much helpful for the researchers. And coming to our education collection, to land on to our core molecular biology and cell biology collection, please drop down the education menu and you can go to biology. Here you can find all our related biology collections, core lab manual and science education collection. I'll just quickly take you to our molecular biology section. So here, these are the chapters that you can see, and this is the video lesson. So as I mentioned, we also have scientist in action videos here, which will discuss some of the key terms and concepts that are being discussed in these video lessons. And if you would like to, select any language of your interest, you can just click onto the language of your interest, and it will play the video in that particular language along with the subtitles. So I'll also show you our science education section. So here we have a cell biology section under advanced biology. You can just click onto this video. Yes, so here, as I mentioned, these are a bit lengthier video than our core video collection, and core video has it exclusively have animations, but here it is a blend of animation and theory. So you can also find timestamps here where if you would like to just play a particular section, you can navigate and play that particular section. And then let me take you to your lab manual. So biology. So we have genetics and cellular processes here. So yes, so the concept video will help the student. They help the student to reinforce the specific concept or the principle of that particular experiment that they are going to run in the laboratory. And the student protocol will have uh, videos on especially on that particular protocol that they are going to run in the laboratory. And then we have instructor prep video which will be helpful for the instructor to give instructions about the material list and how to prep the laboratory and the student for that particular experiment. So apart from all these collection, I would also like to show you one of the richest collection that we have that will be really helpful for teaching. We have faculty resource center here, so where we have our ready to use playlist which can be loaded directly to your LMS. So I will tell you how to reach that. So once you click Faculty Resource Center, you can find videos mapped to your courses and teaching labs. So once you click onto biology section, you can find playlists which are already, pre-made playlists that are ready to load to your LMS. So you can click onto cell biology. So this is a glimpse of our ready to use playlist. It is a comprehensive playlist actually, which you can load with the help of Prerna. She'll be happy to show you. So apart from these, we also have many textbooks that are being mapped, some of the commonly used textbooks that are being mapped. So that to reach that, you can go to biology collection here. And from here, these textbooks are already mapped based on the TOZ of that particular book. And you can make use of this rich resource as well if you would like to integrate those in your courses. And we also have OERs here, open education resource, which are open stacks textbooks from different subjects, so you can make use of these as well. So to conclude, if you would like, if you feel that you have a course outline under syllabi that you feel will be relevant to map using our content from Joe, so you can reach out to Prerna. We have a team of curriculum specialists and a team of subject matter experts who will be taking care of your request. So I'll quickly stop sharing the screen and hand over the session to Prerna. Thank you everyone for your attention, and I'll be happy to take any questions if you have at the end of the session. Over to you, Prerna. Thank you.
Thank you, Preeti. I have shared a course mapping form link, so you can feel free to share your syllabi. Very quickly, in the interest of time, I'm going to just brisk through how the embedding works, how you can make it available for your students. So say, for example, if you're a teaching faculty and a subscribing institute, you can simply go to the video page of your interest. So first, I'm going to show how to have embedding content done on Blackboard LMS. So just click on embed code. This will bring you to the embed page, and you can quickly just select the copy embed code. If you're using Blackboard, what you need to do is on your courses. So from the content, go to the build content section, look at create an item, fill the necessary details, and find the HTML editor. And here what you need to do is just paste this code. Once you save it, just scroll down and click on submit, and this should appear for your students very easily right here. If you are teaching a My Class or something that is associated, if you are going to access My Class, there are very simple ways to go around it. So as a faculty, you will be able to create a My Class. Select create a new class. Once you create a new class, you will see this dialog box open. Fill the required details. So for example, class name, faculty details, instructions to students, if any. The one thing that you should pay attention to is the access type, open or restricted. What it means is if you have access and your institute provides institutional email ID to your students, they just need to log into Jove via their institutional email ID. Or if they're using open domains like Gmail and Yahoo, they can simply, the faculty can simply select open, and the students will be able to join through the open access domains also. Once you create that section, you create a class, you can add playlist, which is compilation of videos. Select the playlist that you would like for your courses and confirm. This adds all the required video lists to your class. Next, if you want to add quizzes for assessments, you can just add the quizzes, either graded, ungraded. It is entirely your choice. And finally, you can just confirm to add the quizzes. Once you add the quizzes, you can click on invite students. And this dialog box will open where you have three options. You can either add a class code. You can share it via the link, which is the most preferred method because you have your internal email listings with students, or you can just upload the email addresses for all of your students. So in the interest of time, I'm going to quickly skip this, which basically summarizes how Jove can help from basic fundamental concept to an application in real-time science. Uh, Next steps would be, if you would like, you can fill that form out that I dropped in the chat and share your syllabi and get a preview of the content we have for you. You can book a demo with me and I can explain even in further detail how to access Jove resources, and we can help you integrate Jove content into your courses. I will quickly hand this over to Tani so that if there are any Q questions, she can move forward with that.
Yes, thank you so much, Prerna, and thank you everybody for giving a wonderful run through the entire content of Jove. So quickly, I would like to mention a few questions that we have just received. So my first question would be directed towards the speakers of the day, Dr. Sudhit and his team. So the question goes like this. So Dr. Sudhit, with so much variety of digital content being available like YouTube and TikTok, which is also coming along with the reduced attention span among students. Could you just give like few quick suggestions and cues for our attendees so that they can make their courses more engaging and interesting for learners, basically?
Yeah, see, we have never faced a problem in terms of short attention span. I mean, our workshops, they start typically 9 o'clock in the morning and they go on till 6 o'clock in the afternoon, I mean, in the evening, and happens over 2 days or 3 days. As long as the content is interesting, the students are going to stick to it. Okay. So what needs to be done is to give a proper context to the students, why they are learning what they are learning, and generally answer the various questions that come to their mind. And if they are asking some questions which we are not able to answer, then acknowledge that and go ahead with it, rather than giving a non-satisfactory kind of an answer. By and large, if these few things are taken care of, students do listen to things, even if they are longer than what the short time span comment would like to us believe.
So thank you so much. So I think in the interest of time, we will be, uh, finishing the Q&A right now. And we have noted down all the questions and we will be getting back to everybody with the answers to their questions. So over to you, Prerna, now. Thank you.
Thank you, everybody. Thank you, Professor Sutir, Dr. Sneha, and Ms. Purnima. This was very insightful, and this was actually very interesting in a way that how hands-on workshop and reinforcing, like the flipped classroom, synchronous, asynchronous, all of these ways and methods, they come together to give a holistic experience for students. I thank everybody who's taken their time out for this session today, and all the registrants and participants will be receiving a recording of this. Thank you very much and have a lovely day ahead.